right. Can everyone hear me okay, hopefully? All right, perfect. So thank you very much for letting me come and give this talk. This will be my first time. Just joined the faculty here in July, so much appreciated. Just a few disclosures, things that are pertinent just on that second part is I have participated in a few BTK inhibitor studies. We're not going to talk specifically about that study, but just relevant for BTKs just in general. So as far as objectives, so I'd like first to discuss, just a general overview of basic concepts of MS pathophysiology and drug development. The idea of this is to sort of provide an update on two of the more major topics that have recently been getting a lot of press in MS. So those are initially the Evobrutinib study, which is a BTK inhibitor. So a lot of you may have seen that had results that came out a few months ago. And then to talk about a newer study on a drug called frexalimab, which is the first anti-CD40 ligand drug that will be studied for MS. So first, I just wanted to mention just, when we think about MS, what are we trying to accomplish? Those of you who are patients certainly think about this a lot. But the overarching goal, if we really simplify things, if we were to look many years into the future, is reducing the development of long-term disability. So more recently, there's been, I think, probably a little bit of graying of the distinction between progressive and relapsing MS. And now we think about disability accumulating really through two mechanisms. So we think about relapse-associated worsening. So what that means is that you have a relapse, you develop some amount of disability. Maybe that will decrease some over time, but most patients will not typically get back to their baseline state. So you can see that represented in the graph on the left side of the screen. So raw is sort of the other term for that. So on the y-axis of that graph, we see disability relative to baseline, and on the x-axis, we see time. And so what you can see shaded in yellow is that there's a relapse, and then a patient will develop an increase in that disability score, and then that may fluctuate. But there's a temporal association there with the symptom change and the relapse happening. For this newer concept, which I know Dr. Corboy had talked about at one of the prior meetings here, is PIRA, or progression independent of relapses. So something that's been more challenging for us to treat. You can think of that akin to progressive MS. So in that case, you can see again that there's a rise in that brown shaded disability, but that is not temporally associated with that yellow relapse. So our job is to try to eliminate this with medications as best as we can. For those of you who have been around the MS story for a while, so we know that we've fortunately had treatments for MS since the early 90s. So if you look at this graph, it's a little bit more detailed than the one that Dr. Corboy had up earlier. But we have injection, infusion, oral therapies, and there's just been a tremendous explosion of this over time. The difference here is just as we look, more towards the right side of the graph, is just that there's an increasing amount of these generic or biosimilar therapies on the market. So as far as concepts for DMT development then, so we're talking about relapses. We know that our current high efficacy therapies, things like Ocrevus, can dramatically reduce relapse rates. We know, again, like with anti-CD20s, that they have dose and duration dependent adverse events, so especially infections, which there's been some discussion about. But we also would think that alternative mechanisms of manipulating the immune system, perhaps in more targeted ways, would be attractive. But we also think of a lot of people that have relapsing MS also will eventually notice changes due to progression. So either with PIRA or that sort of older term of secondary progressive MS. For progressive disease, we have fewer treatments. So Ocrevus, if we think about PPMS, is the only drug that is currently FDA approved. And we have thought, similar to with Dr. Corboy's talk, that there is inflammation that is more sequestered or hidden behind the blood-brain barrier. So different therapies may not be able to access and impact that inflammation as well. So that's where there's always room to try to think through different potential options. And that's where we think of the smoldering inflammation. So immune cells like microglia, for instance, may be important. There we go. Before we walk through two of the more recent studies that have been done, just to put these into context of sort of how far along in the process are we, there's a lot of stages of taking a drug from the lab to clinical practice. So we have tens to hundreds of thousands of compounds and things that are studied in the lab, what we would call a preclinical stage potential drug candidate. We have phase 1 studies, which we're not going to talk about any today, but those are primarily looking at pharmacokinetics, pharmacodynamics, and safety. So those are often small studies. We'll talk about one phase 2 study. So those are typically intended to, in some respects, look at dosing. In MS, we look at MRI efficacy and then also get a safety and some clinical efficacy signal. And then phase 3 studies are the studies that have primary clinical outcomes that are mandated to have approval from the FDA. So we'll first talk about one of the BTK inhibitors, which is Evobrutinib. Evobrutinib works on an enzyme called Bruton tyrosine kinase. So BTK is the acronym there. If you look at this chart, you can see how critical BTK is felt to be in the development and activation of B cells. So again, B cells are part of the adaptive immune system. They are known to be a major driver of inflammation in MS, as well as a number of other inflammatory conditions. So everywhere here that we see a red arrow is a spot that this Bruton tyrosine kinase enzyme is felt to be important. If we think especially about these mature B cells over towards the right side of the screen, and then that activation piece. So this may be a very important way to reduce inflammation in diseases like MS. I had mentioned a little bit earlier, so microglia are what is called an innate immune cell that is resident in the brain and has been emerging as something that may be driving a lot of inflammation in progressive MS. So BTK, if we look at that red box, is known to play a role in creating pro-inflammatory microglia, so microglia that exist in the brain, the spinal cord, and may result in additional inflammatory injury. And those have not been clearly directly targeted by any of our current MS therapies. So as far as BTK and development for MS, those of you who have been to prior research updates will know that there's been quite a few of these drugs that are in different phases of study. There's some pharmacological differences between them as far as how they bond to the BTK enzyme, including covalent or non-covalent bonding, which we don't have to get into, as well as reversibility. But several of these are in phase 3 trials for either progressive or relapsing MS. So to speak specifically about Evobrutinib, because that is the only phase 3 study that has actually been completed and had results, we'll walk through the evolution trials here. So these were set up for relapsing disease, basically to compare Evobrutinib against teriflunomide, which is a Aubagio. So an MS oral therapy that has been out for about 10 years at this point. So how this worked, it was a multi-center study. It was randomized. Patients were essentially put on Evobrutinib and a placebo, or teriflunomide and a placebo. And the reason for that is that Evobrutinib is dosed twice daily. So we wanted to not have patients have a clue of what they were on, just simply based on how often they were taking the medication. And then they were monitored, so they had to be followed to be included in the analysis for 24 weeks. And I'll mention as well here in a second, but a lot of these patients were worldwide, so worldwide inclusion. So as far as who was included, these patients were very similar to our typical phase 3 MS relapsing disease population. So they had to have some degree of clinical stability. You can see their EDSS, so patients with a lot of advanced disability not included. And then as is standard and has come up with some of the other talks, patients had to be under the age of 55. The only major difference between this and our typical study would be some of the exclusion criteria regarding liver function. So there have been some concern from some older studies that these BTK inhibitors may result in some liver toxicity. So there were some additional issues there that these patients who either had prior liver injury or were at risk based on certain conditions were not included in this study. So as far as endpoints, so all of our phase 3 trials for relapsing MS are going to have a primary endpoint of annualized relapse rate. So you basically take all the patients over the course of the study, figure out how many relapses they have, and then put it into an annualized metric. So you get, a sense for how many relapses they may have in a year. There are sometimes some adjustments to that based on baseline factors. And then with studies, we always look at some secondary endpoints that can also give us some clues about, how different things are working. So those, we'll talk through a few of them, but they include MRI outcomes, as well as a newer biomarker, which is called neurofilament light. So these were pretty typical endpoints, as I explained, and the neurofilament light has been increasingly included as a biomarker for MS studies. Demographically, so these, just we're always looking in the studies, do the groups look similar? Because you can imagine we may get a false positive or a false negative study if the groups are dramatically different in things like age or disease activity. These look very much the same. The one interesting piece for us here to think about is just the heavy component of patients from Eastern Europe. So that has increasingly been an attractive place to recruit patients for MS clinical trials just because of the prevalence of the disease, the lower cost, and then some centralization of the health system. So I would say the disadvantage that's pretty clear for that is we already struggle pretty dramatically with being inclusive of different types of racial, ethnic, and diversity in our MS clinical trials. So the fact that those populations are not particularly diverse, if that's where we're going to be running our trials, that is a major issue. And we'll also see that with the frexalimab study here in a few minutes. So primary outcome for this, so again, looking at annualized relapse rate, and again, these evolution studies were just run in parallel. So these, we would expect very similar results because these were essentially the same thing done twice, which is just a way that the FDA likes us to operate, is that unfortunately we did not see a therapeutic benefit from Evobrutinib. So if we compare to teriflunomide, which is probably on the lower end of efficacy of our approved MS medications, we see that the annualized relapse rates were extremely similar, and we did not see what was hoped for, which is that Evobrutinib would lead to a reduction in relapses compared to teriflunomide. We get additional clues, as I was saying, at times from MRI activity. So two ways we can look at that are lesions that are T1 gadolinium enhancing. So those are enhancing lesions that are considered to be actively inflammatory, or new T2 lesions, or T2 lesions on the MRI that are getting bigger. So, if we see a therapy that's more effective, ideally we see that this MRI activity is reduced, which is a great complement to looking just at clinical relapses. Unfortunately, again, we did not see any statistically significant benefit to Evobrutinib. These numbers looked essentially the same between these groups. Neurofilament light, as I explained, is one of the newer markers that's being explored in MS. So what this is, if you look over on the left side of the screen, is neurofilament light is a structural protein. So it is thought to be damaged over time with neurodegenerative and inflammatory processes in MS. So we would expect that level would decrease with an effective therapy. And the way that this has typically been looked at is after about 12 weeks on drug. So they did, in fact, look at week 12 in the evolution studies between the two medications that were trialed, and there was no difference in those metrics. Finally, we will look at confirmed disability progression. So again, what we ultimately care about longer term, do these drugs look different as far as, how much accumulated injury or disability people demonstrate on exam. And again, unfortunately, we did not see any improvement with Evobrutinib compared to teriflunomide. I had mentioned liver toxicity as an issue for Evobrutinib. So when we look at this graph, these ALT and AST numbers are essentially just liver enzymes. So when they are elevated, we get concerned that the liver is being damaged. And you can see that those numbers are higher at several different sort of gradations there for the Evobrutinib group versus teriflunomide. So this would certainly suggest, as has been thought of in prior studies, that there is an increased amount of liver toxicity with Evobrutinib compared to teriflunomide. So as far as, what did we learn from this and what, may we want to not overgeneralize and what's going to happen here in the future? So the study was not set up to sort of do this non-inferiority analysis that Dr. Corboy had mentioned with his discontinuation study. But the way that I would interpret this, just by looking at the numbers, is that Evobrutinib appears to be very similar in efficacy to teriflunomide, but with more toxicity, likely more cost, and a lot less experience. Because as I said, just as a metric, teriflunomide has been out for about 10 years now. So that drug was actually dropped in March. That was announced at one of the investor meetings, so not going to be developed further, unfortunately. So I would say there's quite a bit of caution, as I showed you on one of those very early slides. There's a lot of these BTK inhibitor drugs out there, and this study is only looking at relapsing patients. So I think there is still hope for these several other drugs that they may actually be beneficial in different phases, just because they actually do all have features that may act differently on different sorts of inflammation. So just some caution about sort of throwing out all the BTK inhibitors at once based on this. There was other data about biomarkers that has not been published yet. So brain atrophy, these things called slowly expanding lesions that are probably most pertinent to progressive disease, but that is not available to talk about at the moment. So we'll really need completion of other studies and specific data for progressive disease to really understand, for sure, are these going to work or not. And then we'll move to frexalimab. So frexalimab is a brand new mechanism for treating MS. So it is basically a monoclonal antibody. So in that sense and mechanism similar to things like Ocrevus, but it acts on this thing called CD40L or CD40 ligand. So you can see that on that green cell. That's a T cell. It has CD40 ligand, and it will typically interact with a CD40 on a B cell. So again, these are cells driving inflammation for our MS patients. Beyond the B cells, CD40 is also existent on CNS resident immune cells. So we're thinking about that innate immune system that Dr. Corboy had mentioned. We have not been efficiently targeting in MS. This may impact, that in addition to the adaptive immune system. So sort of a broader mechanism that has not been accomplished, at least with any of our current MS therapies. We know that patients with MS have elevated levels of some of these markers. We know in animal models that blocking this pathway can reduce inflammation. So the hypothesis for this drug is that CD40 ligand-mediated pathways potentiate multiple sclerosis lesions and disease progression. And then we're going to try to block that interaction and see, how patients do. So the phase 2 study for frexalimab, so again, compared to the thousands of patients that were in the Evobrutinib study, this is a smaller study. You can see there that there was about 120 patients that were included, and this was essentially set up with 4 groups. So there was a infusion group, so a high-dose frexalimab, a placebo group that basically got saline instead of frexalimab, so it did get an infusion. A lower dose frexalimab that was injectable, and then a placebo group that got basically a saline injection. And this was a shorter study, so it basically looked at MRI outcomes, which is what most of our phase 2 studies do, and it was over 12 weeks is what we have data for at the moment. So again, the groups look very similar to the typical MS studies that we see. And again, this is for relapsing or active secondary progressive MS. So people have had recent relapses to be included in this study. But again, if we look up at the third category there, so we see again that there is extremely limited diversity in these studies. And again, this is because there's been a shift to doing these in Eastern Europe for a variety of reasons. So as far as outcomes, as opposed to relapses in a phase 2 study, our primary outcome is going to be looking at MRI activity because it's a short study. So we look at basically looking at those enhancing gadolinium enhancing, so inflammatory T1 lesions at week 12, we see that in the pooled placebo group, so that is the high dose and the low dose placebo versus the frexalimab groups, the frexalimab groups have lower numbers of MRI lesions. So you can see that in pooled placebo, we're looking at 3 versus 1.4 if we adjust based on some baseline factors. And if we look at frexalimab, we're looking at 0.4 to 0.2. So a pretty dramatic decrease, which we can also look at here on this slide, where we look just to get a better visual graphically. So on the y-axis, we have mean number of T1 gadolinium enhancing lesions. And then there on the x-axis, we just have our different groups. So we can see that with both of the frexalimab groups, there was a reduction in the number of these lesions at 12 weeks. So this is very promising. The reason you may ask for different doses is we may need this drug to get into the brain to impact the microglia itself, as opposed to things like Ocrevus and Tysabri that can really impact relapses just solely by staying peripherally. So the thought is that if we give more drug, more of it may get into the brain and impact these innate immune pathways, but we're not really looking at progressive disease yet, just with this one study. As far as adverse events, as you might expect, we have more infections in these groups just because we're really blocking different parts of the immune system from functioning normally. But a very short study, so we really need much more data over time with bigger studies, which we'll talk about very briefly, to truly understand what that adverse event profile looks like. So as far as just conclusions for frexalimab, so reduced MRI activity for relapsing MS in a placebo-controlled phase 2 study. Phase 3 studies are going to be critical to confirm these results and also better understand safety. So we will be participating in 2 phase 3 studies here at the University of Colorado in the next several months that these will be starting up. So one will be a phase 3 study essentially comparing frexalimab and again teriflunomide in patients with relapsing MS, and then a different study focused in on that progressive piece. Comparison with placebo in patients with secondary progressive MS. So we are very excited that this is really the first time that we've looked at this specific pathway. So we are hopeful that this will start to act on the biology of MS in a way that we have not accomplished before, especially for progressive disease. So as far as my overall conclusions here for the talk, so the impact of BTK inhibitors in relapsing and progressive MS, in my opinion, is still uncertain. I would not throw everything out just based on this Evobrutinib study, and we'll expect results from the variety of studies over the next 12 to 24 months. Frexalimab represents a novel opportunity for MS treatment, but we have very limited data right now on that, just with this 100-patient study. And then there are a ton of treatments in development that we have not talked about, just due to time limitations, that are also very promising for treating both progressive and relapsing MS. So there's a lot out there, and we're very fortunate to be in that space. So I thank everyone very much for your attention. We'll answer some more questions during the Q&A, and if anyone has anything really specific, you can also always feel free to reach out to me by email. So thank you very much.